Thank you for joining us for the 4th annual AAKP Policy Summit, Patient Voice and Patient Choice. My name is Diana Clynes, and I'm the Executive Director for the American Association of Kidney Patients. Throughout my 16 years with the organization, with all the initiatives that AAKP leads in respect to education, research, patient engagement, and advocacy, I'm especially proud of all the work AAKP has done and continues to do on Capitol Hill to help protect all kidney patients and all living organ donors. Whether you are a person who is insured or uninsured, AAKP believes that as an organ donor, you should be protected from life and disability insurance discrimination. Today, I am pleased to introduce a number of panelists who will present on innovations hitting the national radar, barriers to innovation, patient choice, and access to care. For over 50 years, AAKP, as the nation's largest independent kidney patient organization, has proudly fought for greater patient consumer care choice in treatments. We believe that quality kidney care is defined by treatments and innovations that allow you to be as independent and as healthy as possible to pursue your goals and aspirations, including having a career, owning a home, starting a family, or retiring securely. To begin our panel, I'd like to welcome Mr. Eric Turowski, Vice President of Market Access for Pharmaceuticals. He will speak on barriers to patient choice and access to care. Eric, take it away.
Hi, everyone. I'm Eric Turowski, Vice President of Market Access for Pharmaceuticals. And I hope you're all truly enjoying the content and discussion here today at the Policy Summit. And I on behalf of myself and Aurinia, I appreciate the opportunity to share a little time with you and share a little information with you. I am very much in support of what you guys are attempting and trying to do here in your mission. And I know looking at one of the primary objectives of this Policy Summit is to accelerate the need for policies that honor full consumer choice. And that's something that Aurinia is certainly in full support of, but myself as well, not just professionally in my role as Vice President of Market Access, but also as a person. And we're all a patient at some point in time, and I truly believe that the therapeutic decision should be made between both the patient and the provider. And so appreciate the opportunity to be a part of today, and was hoping that I could share a little bit of information, a brief overview of Pharmaceuticals as an organization, and lupus nephritis, which is a condition that we're currently very much involved in right now. I'll give you a brief background of that for contextual purposes so you can understand some of where we're coming from. Share with you some of the things that we believe get in the way of the objective that I mentioned earlier of allowing patients and consumers to have the therapeutic choice. And then also share some legislative policies that have been introduced and in some situations, in some geographies have been implemented already, but I think need to be broadened, and would love to share that with you and ask for your support. So Pharmaceuticals, we are a small biopharmaceutical company with the mission of changing patients' lives that suffer from autoimmune diseases. That's where we're focused. And we were established in 2013. So we're a young organization. Actually, our commercial organization was stood up basically within the last 12 to 18 months, so during this pandemic era. And although Aurinia itself was established in 2013, our roots go back developmentally on the product that we're working on about 20 years. And so January of this year was an exciting milestone for us and an exciting milestone for patients, where we achieved our first FDA approval for the first approved oral product for the treatment of lupus nephritis. Our organization right now has almost 300 people, and within that we have dedicated individuals for market access, such as myself and my team. We have advocacy, people dedicated to advocacy, and also government affairs and government policy. And the way we approach our strategy is based off of developing very meaningful relationships and being a collaborative partner to many different patient and advocacy organizations, such as AAKP and many others. You can see we're consistently engaged with greater than 50 organizations like yourself. So a little context on lupus nephritis. So I'm not sure those of you in the audience that have familiarity with lupus, but lupus nephritis is a condition, a very serious and often life-threatening condition that affects up to half of patients with a broader, what's called SLE diagnosis of lupus. And the prevalence data is not all that well understood across the US. We believe there's roughly 200,000 to 300,000 patients with SLE or the broader lupus diagnosis. But what we do know is that 1 in every 3 of those have already developed lupus nephritis at the time of their SLE diagnosis. There's a huge slant in this condition towards the female population. In fact, about 90% of patients with LN are female of childbearing age. And there's also a much higher prevalence in the non-white population, so the Black, Asian, and Hispanic communities. I've mentioned that it's a very serious condition and often life-threatening. I wanted to put a little extra context around that for you. Those with lupus nephritis are at a 45 times greater risk of developing kidney failure compared to the general overall lupus population without lupus nephritis. And in addition to being 45 times more likely to have kidney failure, they're at a 5 times greater risk of cardiovascular mortality, and obviously considering those two things, an increased risk of premature death. And it's not hard to understand how those things translate into an economic burden as well for the US healthcare system. In general, the patient population with lupus nephritis have twice the hospitalization rate when they're in the hospital. They're there for on average twice as long. And the costs associated with patients that develop kidney failure is 5 times greater than those that don't. So you can imagine, for a condition like we just spoke about on the last slide there, it's got to be pretty difficult to be a patient living with that when there are no FDA approved therapies. And that was the case up until just this recent December. So it was a short time ago when the first product was approved for LN. And subsequently, within a month of the first one, the second one was approved. And as I mentioned, the product that Aurinia manufactures and offers commercially is the first oral approved for lupus nephritis. And patients want and obviously need access to this new innovation and to new therapies. And in the word bubble there, you can see an example of some of the patient voices once the FDA approval came through. And I think a lot of those comments and quotes, if you quickly glance through them, show examples of true excitement, hope, and hopefully a little bit of relief or a little bit of weight off of their shoulders, as well, as they know now that there are some approved therapies for the condition that they've been suffering with. So when it comes down to it, who should be making the therapeutic decisions? And that sounds to me like somewhat of a silly question, and maybe it does to you as well. It seems like the answer would be obvious to that one, that the healthcare provider and the patient collectively should sit down and talk about the patient's current condition, the benefits and risks of any given therapy, and mutually align on what they believe is the best path forward. I think that just absolutely makes sense. But I think we need to acknowledge, and part of what AAKP is doing in convening this group and talking about these topics is acknowledging and raising some awareness that that's not always who makes the therapeutic decisions. And why is that? What gets in the way of that? There are a host of things that can get in the way of therapeutic choice. I've mentioned 3 of them on the screen here today. One is prior authorizations. I won't go into great detail on that. Many of you are probably familiar with the fact that there is sometimes a large administrative burden, a lot of paperwork and things that have to be fulfilled to the insurance organizations prior to them agreeing to cover the product. So that's something that sometimes gets in the way. Copay accumulators, that is a topic that deserves its own dedicated talk for sure. And if you're not familiar with it, I urge you to go and get a little more familiar with it. Do your Google searches. Essentially what this is even if there is an entity like, say, an Aurinia Pharmaceuticals, who is willing to pay the bill for the portion of the costs that patients are responsible for to their insurance company, the insurance companies that employ these copay accumulators say, thank you very much, we'll certainly take that, but we're not going to let it count towards the patient's deductibles. Once we basically take all the money that whoever it is, the manufacturer in this example, is willing to put forward on the patient's behalf, we're going to still have that patient's deductible starting out at the ground floor, and they're going to have to come out of pocket for the same amount they otherwise would have had to anyway. So that's something that deserves its own talk. But the one that I wanted to spend a little bit more time on here today is step therapy as a tactic. So step therapy, I don't know the familiarity in the room here today, or I guess over video, the familiarity of all of you with step therapy, but I wanted to take a second to give a little bit of an illustration to contextualize what this type of policy that insurance companies utilize is, the type of impact that it can have. So let's just say patient and healthcare professional are in the doctor's office. They do, like we talked about earlier, they talk about the patient's condition, their current status. They talk about the therapeutic options, the benefits, the drawbacks, and ultimately they leave that office visit with a conclusion of the direction that they'd like to go and the therapy that they collectively think is best for the patient. And they send that decision off to the patient's insurance company. And the insurance company in that situation, if they have a step therapy policy in place, would say, essentially, thank you for your opinion on the product that you would like to use. But we at insurance company A, B, or C have decided that before we're willing to cover that product, we want you to first try for a minimum period of time and document that this other product first has been failed, or the patient failed on this first product before we're willing to let you go on to the next step. And in some situations, there's double step edits in place. So after you've tried and had a documented failure on the first medication, we now want you to step up and try for a minimum period of time and have a documented failure of another therapy that we prefer before we'll give you authorization and coverage for the product that you and your patient decided was your first choice all along, right? So step therapy is also commonly referred to as fail first policies. And if I deal with insurance companies and the healthcare economics and our state and U.S. Healthcare every day. And so if I'm putting on the other shoes and I'm trying to think about the growing trends, the rising costs of U.S. healthcare, the fact that we need to control the spend, I understand those things. And could there be a place where step therapy could be implemented in the U.S. healthcare system? Possibly. And I think that a place where it seems to me like it would be fair to employ those tactics would be situations where there are multiple therapies available for a given condition, where multiple therapies have the same mechanism of action. So in the patient's body, they actually work the same way. They've been proven with randomized controlled clinical trials to have a similar therapeutic effect. So patients should expect the same clinical outcome from any of these products, and they're administered the same way. It's not like one is an injection and one is a pill. So if there's that much similarity within a therapeutic class, there's that kind of opportunity for alternatives, then I could see where it could make sense as a means for us to control healthcare spending in the US to employ a tactic like that. But as an example, in lupus nephritis, none of those similarities exist. I told you earlier that until December of this previous year, just a few months ago, there were no therapies that were FDA approved for this condition. There are now 2 that are approved, and they have very different mechanisms of action. They have very different clinical outcomes, both on the efficacy side and on the safety and tolerability side. And they have different modes of administration as well. One is oral and the other is either injected sub-Q or through an IV. And on the right-hand side here, you'll see an example from lupus nephritis, an actual policy that is in place where it says, patient must have had a trial and failure or contraindication of 2 of the following therapies. And there's 3 listed there, right? Mycophenolate mofetil, or that plus tacrolimus, or rituximab. And I will just share with you, all 3 of those options are not FDA approved for the treatment of lupus nephritis. So it's hard to think about this from a patient or a provider's perspective, that you would be asked to try and fail on 2 different therapies, both of which are not even formally approved for the condition that you're trying to treat, before they'll allow you to get coverage of the product that you know or believe to be right from the beginning. And so, it's kind of interesting if you think about the question at the bottom of the screen here. Think about any other industry where they require a very educated professional. We're talking about, doctors here and physicians assistants and people like this that are trained for a long time to think through these decisions and make decisions. What other industry requires their professionals to try something else and fail before they can do what they believe to be correct from the beginning? Would we ever ask an engineer to make sure that they fail before they're able to do, before we give them authorization to the way that they believe to be correct from the beginning? I think it's really interesting when you think about it from that perspective. What can we do about it? What can we stand up and recommend? If we can't outright ban the approach of step therapy, I think that we definitely could do a better job of putting legislation in place that limits the abuse of it. Okay. And there are some examples on the screen here, one at the federal level and one at the state level. At the federal level, there is Senate Bill 464 that has been authored by Senator Lisa Murkowski out of Alaska. And you'll see part of the bill that I think is relevant to the discussion that we have highlighted here today. It says, if patients have already tried and failed on the required step therapy drug, then they should be able to get around the step edit, right, or the step therapy. If the proposed step therapy, if in the situation where there is a step edit, if delayed treatment would cause irreversible consequences, the drug is reasonably expected to be ineffective, or it would lead to severe or irreversible consequences. That should be enough rationale for any provider or patient to be able to go around the insurance company's preferred step therapies. Okay, and like the situation in LN I spoke to earlier, these are serious, life-threatening. This is a serious and life-threatening condition. And when you lose a nephron in your kidney, that is gone forever, right? So any damage to the kidney in that way is irreversible. And so this type of legislation would certainly help patients living with lupus nephritis be able to navigate around some of these step therapy protocols. And at the state level, there's an example here that I've pulled out from the state of Georgia. And it says the required prescription drug is expected to be ineffective based on the known clinical condition of the patient, the known characteristics of the prescription drug regimen. So if the step therapy meets that qualification, then the doctor should be able to get around the step therapy and have access to the therapeutic that they believe is more appropriate for the patient. What I have up on the screen here now is a map. It's actually an interactive map that can be found at the web address listed on the bottom of the screen at steptherapy.com. And it shows you where step therapy legislation to help block the abuse, limit the abuse of step therapy. This shows you where policies like that are in effect already. It shows you where legislation to limit that abuse has been introduced. And then obviously the remainder of states where neither of the two are true at this point. And I'd urge you to get online and educate yourself of, the state where you reside. Where is legislation at on this very important topic? And if you have the opportunity, I'd ask you to contact your local legislators and voice your opinion at the state level and also at the federal level in support of yourself, your own decision-making rights, your family and your friends and patients around the U.S. who are battling diseases with which they would like a certain medication to try and combat that condition. And they're unfortunately having to battle the step therapy requirements. And so at the end of the day, our belief is that, like you, we think that patients and providers should be the ones that make the decisions on what therapies is best for a patient and what approach a patient should take to combating whatever condition they're afflicted with. And we also believe that step therapy, for many of the reasons I shared today, have the ability to really harm patients, if not limited in the abuse. And so our ask, as I just mentioned, is for you to understand the current state of step therapy legislation, both locally and federally. Get in touch with your legislators and voice your opinion on behalf of yourself, on behalf of your friends and family and all the patients out there. And I hope that you enjoy the rest of the day. I appreciate the opportunity to chat with you today, and take care.
Thank you, Eric, for a great presentation. I'd now like to welcome Dr. Andrew Lazar, Chief of the Department of Nephrology at Ahuja Medical Center, who will present on Paradigm Shift, Equitable Access to Genetic Testing. Please go ahead, Dr. Lazar.
Hi, everyone. I want to thank the AAKP for this opportunity to discuss genetics and to discuss racial disparity and what the literature are surrounding them and how they're linked together and what genetics can do to perhaps break down some of that barrier. So I want to thank Natera as well for the opportunity to talk about Renasight. We have this issue in the United States, or certainly the world, where chronic kidney disease is taking off. We have about 30 million in the United States who have chronic kidney disease, and we are going to push 1 million who do have ESRD. But we know that if approximately 13% of our population are Black patients, that about 32% or so are the patients who are on dialysis. So what are the many factors that lead to this extreme disparity for patients who are on dialysis? And yet, once on dialysis, we have literature to show that the Black patients actually have better survival on dialysis, but they're less likely to get transplanted. So that's a problem as well. Like you see here, the incidence of kidney failure is approximately 3.5 times higher for Blacks and 1.5 times higher for Hispanics than for whites. Those on dialysis have a lower socioeconomic state, and unfortunately, racially segregated areas have poorer health outcomes. What are some of those factors? Unfortunately, socioeconomics play a role, lifestyle plays a role, the prevalence of having diabetes and hypertension is also more significant in that population. So it's very complicated, but what's the role of genetics? What's the role that genetics play in one's risk, and then these other factors seem to multiply that risk? So again, when you look at the literature, sadly, socioeconomic status plays a very significant role in why we have a racial disparity of chronic kidney disease and ESRD in the United States and likely in the world. But again, genetic factors likely play a role. One such factor that we'll talk about, apolipoprotein L1, is getting a lot of fanfare because the literature suggests that it's very important and suddenly may be a target of therapy. So we'll talk about that as well. But that's not only more prevalent in the sub-Saharan African patient, but it's also more prevalent in patients in the Hispanic community. So only with doing genetic testing will we learn more as well. So genetic factors. Now, again, most of the diseases that we deal with in nephrology are probably in that polygenic area where one may have a genetic predisposition and they have a second and third hit that play a role. One of the more important genetic factors that have been unearthed is apolipoprotein L1. Now, apolipoprotein L1, this is the rarest of types of medical problems where it is quite common and it has a high medical effect. Now, that's a combination that we don't see very often. And we're learning that it's a fairly prevalent problem, and there are two particular variants, what's called G1 and G2. Now, if a patient has a G1, G1, G2, G2, or a heterozygous G1/G2, it places them at much higher risk for the problems that we see. The problems that we have probably called hypertensive nephrosclerosis, the FSGS that we see, as well as the patients who we find are at greater risk for HIV nephropathy, perhaps SARS-CoV-2 nephropathy. There are data to show that apolipoprotein L1 may place women at greater risk of preeclampsia, but interestingly, not necessarily the apolipoprotein L1 status of the mother, but actually of the child. So there's so much that we have to learn, and in doing genetic testing, as we find more and more patients who have apolipoprotein L1, we can start to target therapies, but we can start to learn more. Now, apolipoprotein L1, and to kind of get back to the other factors, we know that while having two apolipoprotein L1 variants like I described places one at much higher risk, it's not all patients. So 7.5% lifetime risk of ESRD in the African American population, double that for those who have two APOL1 risk alleles. So there are many other factors. And along with socioeconomic, unfortunately, comes likely smoking and other lifestyle habits, and also perhaps the quality of a pregnancy and whether one is born small for gestational age. We know that we have all the nephron number that we will ever have, approximately 7 months in utero, and then it's all downhill from there. So if one is born with fewer nephron number and they hyperfiltrate, the risk of secondary FSGS, and then you throw on a second hit, for example, apolipoprotein L1, you have a higher risk of having these problems. And the literature also suggests lower nephron number at birth, greater risk of obesity and diabetes and hypertension, as well as chronic kidney disease. So in medicine, it's frequent that, yes, we can have a monogenic genetic disease, which is going to lead to chronic kidney disease, ESRD, but maybe even more often, we have a genetic predisposition and those second and third hits that lead to that chronic kidney disease and unfortunately even landing on dialysis. We are in an era now that we have an opportunity to do renal genetic testing, and thankfully this is available for all patients. Now, when I say it's available for all patients, people need access to healthcare, and we certainly have a racial disparity there as well. And that's something that we have to contend with and we have to continue to work on. But I hope that every patient who gets to their primary care physician, to a nephrologist, does have the equal opportunity to be considered for genetic testing. And in doing so, and I have found this personally in my own practice, it gives me an opportunity to unearth the etiology of, let's say, a CKD of unknown cause. It allows me to take a disease that I suspect one has, but to better understand it. What is the mutation? What is the prognosis associated with having that particular mutation? So I may confirm a suspected hereditary cause, or I may even reclassify that. The literature will suggest that I thought that it was FSGS. It turned out to be a collagen 4 disease, or it turned out to be dent disease. I have found a lot of the apolipoprotein L1, and what are the ramifications for patients. On the prognostic side, I may be able to specifically target a therapy for what they have based on what I learned, and I may have an opportunity to talk to the family members, children, the people who are behind the person who, let's say, is sitting on dialysis. When you consider and then make a genetic diagnosis, you're doing it not only for the patient, but for the entire family, and we have to consider that our responsibility. So what is Renasight? Renasight is an opportunity for us to, the way that I frame it when I see a patient, and I don't know why they have CKD, or I put them into this hypertension basket, or I suspect that they have something, but I'm not really 100% certain, and I want to better classify what they have. I have an opportunity now with fair ease to order a test, a gene panel of 385 genes that are associated with the diseases that I don't want to miss. The cystic diseases, the glomerular diseases, the complement diseases, which we are recognizing are a bigger and bigger problem, maybe even associated with IgA nephropathy, the alternate complement pathway. We find that nephrolithiasis, there are some studies that find that as high as 15% of nephrolithiasis can be defined as a monogenic kidney problem for which there may be a specific treatment. So I have an opportunity, the way I tell patients, to look under another 385 stones and unearth what the true cause of their disease is. And when I do that with this particular Renasight panel, I know that I have a team on the other end who are going to use a number of complex tools to do this next generation sequencing. And they know that certain genes, for example, the PKD1 gene may be a little bit tougher to look at, and they'll use certain techniques for that gene and other techniques for others. So, and when you look at the quality of the study, they have greater than 99% detection rates across the genes that they have chosen. And remember, how do they come up with 385? We have 20,000 genes, the human genome, 4,100 genes associated with Mendelian human disease, 625 and counting of those, your nephrome that is associated with, or that are associated with kidney disease. Take those 625, come down to 385 to really improve our yield of the problems that we don't want to miss. And now we have an opportunity to miss fewer diagnoses. So when you look at the literature, and there are more and more literature that we're seeing in our top tier journals, that this particular trial, for example, this was in January of 2019, Groupman and team at Columbia University looked at 3,300 patients, and they, of whom 65% were on dialysis, and they found that upwards of 9.3% they could make a monogenic kidney disease etiology diagnosis. And when you consider those patients for whom they made a genetic diagnosis, 89% of those, there was clinical implication for that genetic finding, whether it was confirming a suspected hereditary cause, which may then help talk to family, it may help with prognosis, it may have been, again, the reclassification of disease. We thought that it was X, it turned out to be Y, very common, collagen 4 diseases, and now we may have treatments for collagen 4 diseases. So we have to make these diagnoses, and in 89% of patients, there were clinical implications. And when you look at Renasight, and perhaps 7,000 or 8,000 of these tests have been done now, and you look at the first 1,000 or so, 70% identified a positive or a carrier state. So it's not particularly uncommon, and when you consider the yield, when you look at the literature of genetic kidney disease findings sitting in our clinic for those hypertensions that we don't know the cause, or those who have proteinuria, hematuria, they've had a negative cysto and you don't really know the cause. The yield in some can be high as 37%, on the low side here, 9.3%, but a fairly high yield in determining what is the true etiology of their disease. And of the first 1,000, Renasight has found this, 75% positive or carrier. And the types of problems that we have talked about, PKD, the apolipoprotein L1, amyloid, which was a surprise. And now we know TTR amyloid could be a bigger kidney problem, not only cardiac, as well as collagen 4. Case example, consider a 31-year-old woman, proteinuria during pregnancy, hypertension. She's thrown into that hypertension basket. She sadly is now on dialysis. You do a genetic test and you do find a TTR amyloid. Now, what are the implications? Well, her risk of heart failure, we have to look a little bit closer perhaps. She's, I hope, getting ready for transplant. And we find apolipoprotein L1. And now we know that there may be implications. It seems that the donor, it may be particularly important, but some newer literature may suggest that the apolipoprotein L1 status for the recipient may also be very important. The results of the Apollo study will help unearth that a little bit more. But these are the types of results that are very important for your patient and the family members. So genetic testing, I find it in my office to be very easy. I know that Natera has a portal where you can order tests. I personally have pre-printed sheets. As the patient's leaving, I find it easy that I have them fill out the top and sign it. I take care of the rest. Something is sent to the patient, they take care of it. I do primarily saliva kits, but also blood can be done. I know that Natera can send people even to homes to have those blood tests done. And I have found that Natera works with my patients if we have issues with cost, to at times bring that cost all the way down to zero, at times $99 or $149, depending on income. Now, what is GINA? I often have to have this discussion. Patients are concerned, should I do genetic testing? I may unearth that I have a problem. What are the implications going to be? Health insurance, employment discrimination. This act should protect patients from this problem. Now, but I've learned that it does not cover life insurance, which is essentially a private product, long-term care insurance, and disability. I had just learned recently that individuals working for a company with fewer than 15 employees, it may not protect them. This is not my area of expertise, but something that one should be aware of and something that we should talk to the patients about. So in summary, we have an opportunity now in our clinic with relative ease and a reasonable cost to look at 385 genes and start to unearth the etiology of what the disease really is. And I look at it in terms of what am I missing in my clinic? What am I missing in the dialysis unit? What am I missing on the transplant waiting list? And there are implications for the patient and there are implications for the family. And when you consider the yield relative to other tests that we often do, the yield I consider high. And it's an opportunity for me to then also enroll patients in trials, really feel that I'm doing the best that I could do for patients. So I want to thank everyone. I want to thank the AAKP. One can visit natera.com/Renasight to learn more, can send an email should you be interested. And certainly that there are representatives in your area who can help as well. Thank you very much.
Thank you, Dr. Lazar, for speaking on such an important topic. I'd now like to introduce Ms. Tricia Brooks, Vice President of Government Affairs and Policy at Ardelyx Pharmaceuticals. Mrs. Brooks' presentation is entitled, New Entry to Existing Market, Do Policies Support Innovation? Welcome, Mrs. Brooks.
Hello, and thank you to AAKP for inviting us to join this session today. My name is Tricia Brooks, and I'm the Vice President of Government Affairs and Policy at Ardelyx. Ardelyx is an emerging company founded in 2007. I think that actually speaks volumes to this session title. We are currently waiting for an approval of our first marketed product here in the U.S. We are committed to improving the lives of patients with kidney and cardiorenal disease by discovering and developing novel and targeted therapies to advance patient care. With approximately 250 employees, we stand ready to change patient care with those living living on dialysis. And I say this as someone whose father is currently on dialysis. I know it's not the answer, but it brings us closer to making his life a little bit more livable. Now it is my honor to introduce Dr. Laura Williams, who is our Senior Vice President of Patient Advocacy. She brings more than 25 years of clinical development to Ardelyx and actually leads our patient advocacy from a science-based, patient-first point of view.
Thanks, Tricia. I'm excited to discuss Ardelyx's pipeline because we believe it has the potential to address significant unmet medical need in a population, most of which are disproportionately affected with chronic kidney disease and cardiovascular disease. Our research expertise in biological mechanisms and pathways has enabled us to have a pipeline that's targeted, has a number of first-in-class drug candidates that are designed to address significant unmet medical needs. Our most advanced asset is tenapanor. It's our lead candidate, and it's for the control of serum phosphorus in adult patients with chronic kidney disease on dialysis. And we'll discuss that a little bit more in just a few minutes. We also have programs that address hyperkalemia, metabolic acidosis, and a vast discovery platform that fuels additional pipeline programs. First, I must address a little bit of information in terms of phosphorus control and our efforts to innovate in this space to address the significant unmet medical need. I think it's important to note that our kidneys are the main regulator of phosphorus homeostasis or control, excreting about 2/3 of our daily phosphorus intake. And so as our kidneys function fails, serum phosphorus levels rise, and that rise is associated with increased morbidity and mortality, as this slide depicts. In the graph shown here, as phosphorus levels increase above the reference range, which is noted in gray, there is an associated significant increase in the risk of death, with a relative risk ranging from 7% all the way to 102%, depending on the increase in the level of serum phosphorus. There's also a relative risk of increased hospitalizations by up to 38%. The KDIGO guidelines, which were published in 2017, recommend that we lower elevated phosphate levels toward the normal range. And in adults, that is 2.5 to 4.5 milligrams per deciliter. It is important to note, though, that despite all efforts from both a patient as well as healthcare provider perspective, the majority of patients are unable to consistently maintain their target phosphorus levels with the currently available therapies. As a matter of fact, our DOPS data, which spans from 2015 to 2019, looking at patients on dialysis, has noted that 77% of patients treated with phosphate binders were actually unable to consistently maintain their phosphorus levels less than 5.5 milligrams per deciliter over a 6-month period. Now, tenapanor is our investigational first-in-class non-binder phosphate absorption inhibitor that is our lead candidate. And this slide describes its mechanism of action. First and foremost, it targets the primary pathway of phosphate absorption by inhibiting the sodium-hydrogen exchange, thereby reducing the sodium absorption, resulting in modest intracellular proton retention. What that does is it subsequently blocks the absorption of phosphate through the paracellular pathway. And the dose is very simple, 1 pill twice per day. Our clinical data package for tenapanor is robust, having dosed more than 400 patients, healthy volunteer patients, and more than 1,000 patients with chronic kidney disease. What's depicted on the slide here is a number of our phase 3 studies, some of them short-term, some of them long-term, some of them extensions, and then another study called Amplify that looks at additional data with tenapanor and phosphate binders. In each of these studies, we met both our primary and secondary endpoints, or key secondary endpoints, and were able to demonstrate an increased percentage of patients achieving goal as it relates to phosphorus levels or serum phosphorus levels. And as Tricia noted, this is our lead candidate, and our PDUFA date for this approval is July 29th of this year. I think one of the most exciting things from a patient advocacy standpoint is really being able to incorporate the patient's voice in our clinical trials. And this slide depicts an example of such efforts. We have included patient-reported outcome measures in our clinical trial in one of the ongoing studies called Optimize. And what's listed here is a number of questions that really depict the voice of the patient. Things like how they would characterize their feelings about phosphorus management during the study compared to how they felt with relationship to phosphorus management before the study. Things like, what do they think? What changes most likely contributed to the change in how they felt with their phosphorus management during the study? So there are a number of questions here that, again, really outline sort of the patient's voice in clinical trials and how we can really enhance that voice to improve our product. I like the last question, which says, which best describes how you feel about the change in one of the effects that we see in the study, which is bowel movements, negative, neutral, or positive. Again, all of these questions are listed in a way that it enhances our understanding of what the patients feel, not only about the treatment, but about their disease and about the management of their disease. So in our own way, we've really tried to bring about an innovative change to this space. I think one of the key questions is, how will that innovation be supported? With that, I will turn it back to Tricia to wrap things up.
Thank you, Laura. So as we talk about policy changes and what policy changes can be made to support innovation and patient access, we often think about these very sexy moves to gene therapy and breakthrough cures. But quite frankly, innovation changes are what matters to an individual. I was very fortunate to work with Christopher and Dana Reeve, and for someone living as a ventilated quadriplegic, it wouldn't necessarily be about walking. It might be about being off a ventilator or getting the movement in their hand or bowel bladder back. For my father on dialysis, it may be about not having constipation during or not having to have fluid restrictions. For each one of us, having what makes our life a little bit easier to manage, having tools to manage our life is true innovation. Being able to live our life is innovation. And so being able to incorporate patient-reported outcomes and what matters to an individual to live their life and prioritize being able to coach their kids' soccer team and not worry about having hypoglycemia if you have diabetes. I think these are things that matter to individuals. So during 21st Century Cures, it required FDA to issue guidance on how they would incorporate patient-reported outcomes into the drug development process. And so what are the unmet needs in somebody's disease life cycle? What are the functional improvements that could be considered innovative? So it's not just ease of use, but it's ease of compliance. It would be important for people in a chronic disease management state. So I really do believe that as a policy entity, how do we ensure that the voice of the patient is actually brought to bear when we see the true definition of what is innovation to an individual? We also look at reimbursement and what is going to spur the continued investment into this space. And so I understand when we look at bundled payments and the need for equity and access to the full life cycle management or or the full life cycle of care for an individual on dialysis? How do we ensure that what was the idea of implementation is actually executed in a way that people have access to what they need and ongoing investment into the disease state? So looking at ways to support all of the aspects of the care and management. And then finally, how do we ensure that we include patients in the dialogue around need for sustainable healthcare ecosystem and what that looks like, not just in basic science, translational science, but the full life cycle management and that ecosystem and what that health and management looks like. We are unique at Ardelyx, and we stand alone right now as a company that continues to be funded for this piece of innovation, but it is running dry, and we want to make sure that we continue this dialogue. And that's important for us to be at the table and having individuals on our management team continue to have this dialogue. So we thank you very much for continuing to take your time to go and meet with your members of Congress. And finally, as all of you return to your days of coming to Washington and meeting with your members of Congress virtually and in person, just know there is no higher priority for a member of Congress than to hear from their constituents. So please bring your stories, your experience, and your desire to see more investment in this disease space and the entire ecosystem of healthcare delivery, access, and innovation. And we thank you. Thank you to AAKP. Thank you to Dr. Laura for joining me, and thank you for inviting us to join you today.
Thank you for a great presentation. I would now like to welcome our final speaker, Mr. Shawn Gage, founder and director of clinical operations for InnAVasc Medical Incorporated. Mr. Gage's presentation is entitled An Innovative Solution for Hemodialysis Patients, Challenges in Delivering a New Technology to Patients That Need It the Most. Thank you, Mr. Gage. You may begin.
Hey, everybody. Thanks a lot for having me at this meeting. I think this is a really important meeting for us all to learn more about this process. And I hope to be able to, through this talk, help you to learn a little bit more and bring you into some of the struggles and challenges that we have in industry, trying to make things better for patients. So today I'm going to talk about what I think is an innovative solution for patients with hemodialysis. But again, some of the struggles that we have of trying to bring these technologies to people that really need them, particularly in the hemodialysis space. So you all know about kidney disease here. And this affects a lot of people around the world. By latest statistics, about 2.5 million people worldwide are affected by end-stage kidney disease. Just here in the United States, of the 800,000 or so patients that we have that have end-stage kidney disease, about 500,000 of those patients require hemodialysis. We all know this is a life-sustaining therapy. They need to have their treatment done at least 3 times a week. But if somebody is on home hemodialysis, that may be as many as 5 or 6 times a week. And it requires needle access to a graft or a fistula if you're not using a catheter for hemodialysis. Unfortunately, AV access has its problems. And in addition to the technical problems, there's a lot of issues with fear, needle fear, cannulation challenges, needle injuries, material breakdown. Most of these grafts that we use nowadays were actually never engineered to be stuck with dialysis needles. There's issues with bleeding. There's issues with stenosis or a narrowing of the outflow vein. Issues with thrombosis or clotting. That happens oftentimes when that outflow vein gets narrowed. They fail. They become infected. At this point, we spend at least $3 billion a year on just managing vascular access malfunctions and complications. That's not even the cost that we pay for managing end-stage kidney disease as a whole. So we've been using the current modes of hemodialysis access like fistulas and grafts and catheters for over 50 years now. And over that time, we've tried to innovate around some of the challenges that we have with hemodialysis access, like clotting or bleeding. And we've done things like we've added medicines to try to reduce clotting. We've tried to modify the outflow in the turbulence that the high flow blood has on the outflow vein. We've tried to do things like improve the thickness or the sealing of grafts to improve bleeding. We've tried to even use cell therapies or gene therapies to modify the way that the veins manage the high flow stresses of the blood from AV access. But what we haven't done over the last 50 years is there's not been a concerted effort to try to manage issues like this. These are some of my patients that have suffered what we call back wall injuries from the needle. As you see up there in the top panel, it's very easy for the needle to puncture through the front wall as it's intended, but it's actually quite easy for that needle to continue on through the back wall to cause these types of very painful and complicated issues with our patients, with large collections of blood under their skin that have to be managed in the operating room. And this is just a little bit more detail about what that looks like and how it happens. With standard grafts, we poke the needle through the front wall. And as the needle makes contact with the front wall, it starts to deflect inward towards the middle lumen of the graft before it actually punctures through. And by the time that needle makes it through the graft wall, you can see how close it is to the back wall of the graft. It's nearly 3/4 of the way through the lumen of the graft. And so that forward momentum makes it very easy for that needle to travel through the back wall and cause needle-related injuries. As you can see here, the needle is coursing through the back wall. So now when the needle is removed, not only is there bleeding from the front wall where we would expect it to be, but now there's bleeding from the back wall that's much more difficult to manage or to control because we can't apply pressure in that area. And over the years, there's not been a concerted effort to address issues like this, from individual needle puncture or vascular sheath puncture infections that can lead to serious complications and bleeding. And there have not been any advancements to try to improve upon graft degradation or bulging from the graft, or we call them pseudoaneurysms. As I mentioned earlier, the grafts that we use today were never intended or engineered to be stuck with a dialysis needle. They were actually engineered to be blood vessel replacements, like peripheral arterial bypass grafts, never meant to be instrumented after they were placed. And you can see from the images below what happens to those grafts over time. On the left and the right side of the screen, these are my patients' pictures of their grafts where they've been stuck with needles over and over for dialysis. And just below them is a corresponding image of their angiogram, where we inject dye into the graft to look to see for any problems. And you can see in the middle aspect of the graft on the left at the bottom, there is normal looking graft there where we're not sticking with the needle. However, what you can see just beyond that on either side is there's actually no graft remaining in that area. It's blood swirling outside of the graft, being contained only by your skin and your fat layers. And why does that happen? Well, if you look at the graft at the image at the bottom of the screen, you can see that's a graft that's been removed from a patient who has had a pseudoaneurysm, just like I'm showing on your screen. And this is what a graft looks like. It looks like Swiss cheese. It essentially has no integrity, and the blood is no longer flowing within the lumen of it. And you see the graft image next to that. That's what happens on the benchtop when you poke a dialysis needle into a regular dialysis graft. Those holes don't close up again. And so we have to rely on the magic of the clotting cascade for our bodies to plug up those holes so that they don't continue to bleed. And over time, that can be less and less stable. And finally, those complications that I've talked about can lead to even more serious complications that we refer to as fatal vascular access hemorrhage. These are the types of complications that should absolutely never happen. As you can see in panel C, this patient had a dialysis graft that looked fairly harmless from the outside. But if you look at panel D, which is the inside image of the graft, you can see that not only was there a material failure here, but there was a failure in the care of the patient, because these types of injuries happen. They can happen with repetitive cannulation over and over again, even if the graft is used correctly and punctured in the correct way. But oftentimes we'll find that areas are punctured over and over again in the exact same spot, and it can dramatically accelerate graft degradation and lead to these types of dangerous complications where people bleed to death in their own homes. This is something, again, that should never happen when patients are trying to receive the type of renal replacement therapy or kidney replacement therapy that they have to sustain life. They should not lose their life during the process of that. So one day in the operating room, my partner and I, Jeff, were taking care of a patient who had a complication with their dialysis graft. And we thought, you know what, there's got to be a better way. We can't continue to fix these problems week after week after week after we've already spent a lot of effort trying to make their dialysis access work to begin with, only for them to come back a few weeks later or a month later with a large injury of their graft that lands them back in the hospital, requires them to go back to the operating room, and all the while the patient is suffering. So after that case, we actually were able to get out and get lunch. Of course, it was about 3 p.m. that day, but we got to get out to eat, and we continued to talk about this. And so we started to make some sketches. We had some ideas. And what did we have? We didn't have a piece of paper with us. We had a napkin. We were in the cafeteria. And so this is the actual napkin. This is how it happens, right? So this was the genesis of the idea of a graft that I'm going to talk about briefly in this talk. We wanted to develop a graft that could protect patients from needle injuries. You could poke the needle into the top of the graft the way that we typically do so that you can gain access to the bloodstream, but we wanted to make it in a way that the puncture site was durable and resistant to bleeding, and that the back and the side walls were preventative from the needle puncturing through the back or the side walls. That leads to many of the complications that we fix often in the operating room. So this is a really long story, and because it's a long story, it's taken 10 or 11 years to get to where I am today from that initial drawing. And so I'm going to touch on the high points. I'm going to fast forward through several areas, because at the end here, I want to talk a little bit about some of the other challenges that we have outside of just development of a new technology. And so I wanted to be able to at least paint the picture for you of the process that we've gone through. And so in short, at that point, we decided to file what's called an invention disclosure form with the university. That's the process that we follow. And that was something that neither of us had experienced in the past, but we knew it was something we had to do to get us started. Skipping ahead many months, eventually Duke agreed to support the idea. This was something that Duke felt had significant value. This took months and weeks of explaining to folks who don't practice medicine what the value is and what the clinical need was. So essentially what that meant is that Duke would supply patent attorneys for us so that we could they could help us protect the idea, because without protection, it's difficult to do anything with your idea going forward. What that meant is that Duke held an initial stake in the company. Again, they were able to put up the initial funding to help us get started. Patent costs are not cheap. So now where do we go from here? Do we do further patents around this to protect this further? Do we start prototyping? Do we need funding? Do we need to talk to the FDA? What are the competing technologies in the landscape? Do we need to do experiments? Do we need to start a company? Do we need to do a business model? What's a business model? Do we need people to help us with engineering? Do we need to license the technology? Well, the answer is all of this stuff. This all needs to happen, and it really all needs to happen essentially at the same time. So, of course, this is a very overwhelming time, particularly for somebody who trained for their whole career to practice medicine and to take care of humans, not to develop new devices, not to develop business plans, not to figure out how to prototype, or to start a company. So again, skipping forward here, there was a lot of progress. We started with a drawing on a napkin. That drawing became a little bit more sophisticated and more detailed. That led to additional patents, patent explanations, and patent drawings. And that led to some early prototypes. And yeah, these prototypes actually started from materials that I had in the lab or in the garage to be able to develop an initial proof of concept, to be able to continue to iterate on the ideas and to explain to the Office of License and Patents and Ventures at Duke what we were trying to do and how we were trying to accomplish that. Continuing to fast forward, we decided to form a company. And why do we form a company? Well, there's lots of reasons to form a company, but in short, really, this is for legal protection, and this is really for a vehicle for funding going forward. You can't do anything without money, and in order for us to continue to progress, we needed to be able to have a vehicle for fundraising and to develop a value stake in the company. So as we continue to move forward, we've had a pretty significant evolution of the design of our device, and we think that we came up with a product that was a solution to the problem that we first identified in the operating room over and over again, something that was continually hurting our patients and causing them to suffer in the hospital. So that brings us to today, the InnAVasc graft. And although there are many benefits that we believe that our graft has the potential to solve, we boil it down to really three main issues that our graft can solve for patients on hemodialysis. Number one is the back and sidewall injuries that we discussed earlier, with the needle poking through the back of the graft or the side of the graft. Number two is excessive bleeding. And number three, particularly for patients at home, is to address the lack of cannulation confidence and anxiety around accessing your graft if you're going to be performing dialysis at home. And to date, there's no hemodialysis access that has been developed specifically to address all of these issues in one graft. So just taking you through each one of these, on the back sidewall injury perspective, what we've done is we've incorporated a back and sidewall plate that makes it impossible for the needle to puncture through the back or the sidewall as long as you puncture in the dedicated cannulation zone. And so that benefit is seen here when the needle tries to pass through the back wall of the graft, it is prevented by the back plate. No needle-related injuries on the back and sidewall. Excessive bleeding. We've talked earlier about how when the needle punctures through the front wall and then the back wall, you now have two zones from bleeding to occur, one of them that you can't control because it's on the back side of the graft. So with the InnAVasc graft, we've developed this technology so that the cannulation zones, or the cannulation pods, as we refer to them as, are truly self-sealing when the needle is removed. As you can see there with the yellow circle, following needle removal, that area seals up completely and does not allow blood to escape from the top side of the graft. And that leads to protection from bleeding. Not only is it protection from bleeding, but because it's self-sealing, that means you can use this graft immediately. No longer do you have to wait 4, 5, 6 weeks while the graft is healing and scarring into your tissue before you can use the graft for dialysis safely. And to address the lack of cannulation confidence, again, particularly for those patients who are on home dialysis, it can be overwhelming to be able to have to care for yourself now at home and have to worry about, can I access my graft? Can I even puncture it in the right spot? And when I do, is it going to bleed or am I going to miss so I can't receive my treatment? So we've developed the InnAVasc graft with two targeted zones to assure safe and reliable access with the dialysis needle. Now, unfortunately, they don't come with these fancy targets on them, but this is to make the point that when you are palpating or feeling for the graft cannulation zone under your skin, you can feel these raised cannulation indicator ovals that allow for you to safely access the graft in the correct zone so that when you puncture the needle into the graft, it's impossible for the needle to exit the graft out the back or the sidewall, therefore guaranteeing your access to receive your dialysis treatment. This makes it safe and easy and hopefully anxiety-free for those patients and the caregivers trying to perform hemodialysis at home. And although this device was developed initially with the intent to prevent needle-related injuries or complications, it became pretty evident immediately after the development that, hey, if this is making the device easier to use, safer to use for those technicians and nurses that are trained to use the device, what about people and the caregivers that are poised to receive dialysis at home? And so with the way that this device is developed, and what we've just described, it would seem that it would make it easier and more anxiety-free for patients and caregivers to use at home. And with the executive order that was put into place a few years ago, it has started to lower many of the barriers that there have been historically to receiving home hemodialysis. But unfortunately, the biggest barrier that it does not address at all is fear and anxiety from the patient perspective, of fear of self-cannulation, fear of bleeding from those complications that can occur from needle injuries, bleeding from accidental needle dislodgement, and then again, the fear of not being able to access your graft on a reliable basis to receive your dialysis treatment. And this isn't just our opinion. Those in the dialysis community have recognized this issue. And not only in the dialysis organizations, but Ms. Gedney from Home Dialyzers is a home dialysis patient. These are real fears that not only she has had to deal with, but many of her constituents and friends alike. So why do we care about home hemodialysis? Well, one of the big benefits is that this can be performed independently and at home. No longer do you have to rely on transportation to get to and from dialysis. But one of the most significant benefits really is the ability for better health outcomes. And that really comes from more frequent dialysis treatments. When you receive dialysis 5 or 6 times a week, as opposed to the standard 3 times a week, you can do shorter sessions and take off less fluids and less toxins at a time, so that it gives you more of a typical function that your kidneys do. And so you have less fluid swings, which reduces any blood pressure changes or massive swings in blood pressure. Patients feel better. It's better for your heart. It's better for your nutrition. And patients tend to have better quality of life. This also reduces mortality. And in particular to this period of time, not having to go to a dialysis unit really reduces your exposure to other communicable diseases that you may be exposed to in the dialysis unit. So what are others doing around the world in terms of home hemodialysis? Well, countries like New Zealand are doing a great job. They've got about 18% of their kidney failure patients utilizing dialysis at home. Australia is close to 10%, and the UK is close to 10%. Canada is about 6% of their patients using home dialysis. But in Europe and the United States, we're less than 3%. In the United States, we're actually at about approximately 2%. So of all of the patients that have kidney failure each year, and as of a few years ago, there were about 750,000 patients with end-stage kidney disease in the United States. 7% of those using peritoneal dialysis, 30% using or had a functioning kidney transplant, and 63% are using hemodialysis as their mode for renal replacement therapy. 98% of those patients are receiving dialysis in center, and only 2% of those patients are receiving dialysis at home. And that really only equates to about 12 to 15,000 patients in the entire country that are utilizing home hemodialysis. And what does that look like for patients that are new to kidney failure? We have about 120,000 patients each year that are diagnosed with end-stage kidney disease. And of those 120,000 patients, about 13,000 end up utilizing home dialysis. Now that means home dialysis means peritoneal dialysis at home and hemodialysis. And as you can see from this graph, only about 4% of those patients, which really only equates to about 500 patients each year, are utilizing home hemodialysis as their initial means for renal replacement therapy. 500 patients out of approximately 13,000. Pretty cool, you say, right? We have a graft that that has the potential to provide safe hemodialysis that prevents bleeding, that prevents needle-related complications, and makes it easier to use and safer to use for patients doing home hemodialysis. Well, we think so too. However, what really matters is how cool the FDA thinks it is. And by cool, I mean how safe and effective does the FDA think that your device is? And this is the part that I really want to get into some of the challenges that we face in the pathway to bringing technologies to patients. Outside of all of the other funding and prototyping and manufacturing and building that we have to do, one of the big main end goals is to make sure that your device is deemed safe and effective by the FDA so that it can actually go to market and help patients. So as part of this process, we have to figure out what kind of device classification is the device in. We have to consider the safety and effectiveness of things outside of just the performance in patients, but how it's manufactured. What's the biocompatibility? Will it cause infections or complications with patients in that way? Are the device specs able to be replicated on a regular basis? And we have to verify, we have to validate all of these specs. And then what type of pathway? Is it going to be what we call the 510K pathway, which is oftentimes utilized for devices that have predicates that come before them? So in this case, there are other dialysis grafts that are out there on the market. And so thankfully, we're able to utilize a 510K pathway that tends to be more abbreviated and less expensive than the PMA route. Do we require preclinical testing? Do you have to do tests in animals before you get to humans? And if you have to do a human trial, what type of a clinical trial? What's one of the first main goals with dealing with the FDA? Well, we have to get to what's called IDE approval. This is investigational device exemption. And essentially what this means is this is permission from the FDA to be able to use your device in a clinical trial testing under their observation in humans. And that means that you have to do a lot of work to get there. We have to determine, are we doing animal work? In our case, we had to do some animal work to make sure that the device was safe and effective in animals first. And in some cases, for some devices, it's none. Some cases, it's some. And in some cases, it's a full complement of years-long or months-long animal work that is extremely expensive and time-consuming. And you can't move to the next step until that's done. Then we have to talk about what's the clinical study going to look like? What's the human study going to look like? And how many patients do we need? Is it going to be an initial feasibility study for some early testing with the intent to make changes to the device to then go on to a final pivotal study? Or can you just move on to a pivotal study from the beginning? How many patients are we going to need? Is that going to be based on statistics? Is it going to be based on FDA's gut feeling? It depends on the device. It depends on the history of the device and other predicate devices. Are you going to compare to historical data? What's in the literature? Or are you going to have what's called a control and do a head-to-head study and a randomized study? We also have to make sure that we agree on the device manufacturing requirements. These are per the ISO standards. And we have to agree on different types of biocompatibility testing. Are we going to do all of it, or we have to do some of it? And it really just all depends. But these are parts of the IDE requirements that we have to be able to have locked down and completed before we can ask FDA, can we do a human clinical study? I want to talk about CMS as well. These are working with CMS and CMS coverage is something that's very important, even with investigational devices. This is something that early on I would not have thought that had to be such a big part of the story while we're doing our investigative work in clinical studies. But there's lots of reasons that you would want to establish a relationship with CMS while you're working on a new device. Some of this is just establishing a reimbursement pathway. Is there already one in place? Do you have to develop a new pathway? Are there codes currently that you can use, or do you have to develop new codes? And so these are all very important questions that really do need to be addressed early on in the process. However, from a clinical study perspective, there are CMS coverage considerations. And typically we talk of those coverages in terms of Category A designation coverage or Category B designation coverage. And actually, FDA is the agency that sets the risk level, which determines whether you're a Category A or a Category B device. If you're a Category A device, it typically means that there's less information about your type of device, and it's considered a higher risk device. Therefore, that means that when you are granted Category A coverage, the procedure that's required to implant your device would be covered. However, the device itself, being that it's still investigational and of a higher risk, is not covered. So essentially, you as the company are providing the device to the patient and the hospital for use in the study. When you get to a point that there's more information around your device and it's considered to be lower risk at that point by the FDA, then you could be granted Category B designation. And what's important about this is that now the procedure to implant your device is not only covered, but your device, your investigational device, is actually covered as well, which means that as a small company, you can start to develop some very small revenue from your devices, which is very important for a small company who has less funding at their disposal. And what is really important about this is that if you don't have coverage from CMS for your investigational study, this means that either the patient or the company is on the hook to pay for everything involved with the patient's care. And of course, we never want to put the patient in that situation at all. And for a company, it's something that, again, from a funding perspective, most small biotechnology companies do not have enough funding at their disposal to pay for all of the medical services for all patients in their clinical trials. What's really complicated about all of this is that historically CMS and FDA do not typically align on their requirements for the clinical studies. You have to agree to a set of requirements from FDA for your protocol and all the other IDE testing that we just spoke of. And those requirements may be completely or at least somewhat different than what CMS requires in order to grant you coverage for your clinical study. What's important about this is that it's important to establish relationships with both organizations early. I think one of the lessons learned, however, from our perspective, is that establishing that CMS connection and engagement even earlier as you're developing your protocols for your clinical trial is extremely important. It will help to reduce any hangups or delays later on as you negotiate with CMS about coverage. So all this to say that there's a lot of planning that's required when you start to get to the point of thinking about your clinical trials. And what are the reasons for your clinical trial? Is it strictly for regulatory clearance, meaning to have FDA approve and clear your device so it can be on the market? Is it for reimbursement purposes so that you can develop new codes and generate new data to improve your value or reimbursement potential? Or is it around marketing to improve the value of your technology or your company? And the answer is really all of these are very important. And again, it's becoming increasingly important to communicate with both FDA and CMS upfront to ensure that you have all the right requirements checked on both sides before you start your clinical trial and develop your clinical trial protocols. Clinical trials are very expensive. And again, all the more reason to reduce delays as much as possible. And why are clinical trials so expensive? Well, you have to figure in the cost of the devices. How much does it cost to manufacture? All of that process around the development and the validation of all of that is extremely expensive. You have to pay for the cost of the performing of the procedure, all of the follow-up visits and tests. You have to pay for the investigators and the coordinators. You have to pay for all the training and the monitoring and the data management. If you require any recruitment or advertising or patient reimbursement. And then there's the study management, the whole team of people that it requires for you to conduct a clinical trial requires lots of people with many years of experience to be able to keep everything straight and to make sure that we're doing everything safely for the patients and so that we can move forward towards our regulatory clearance and ultimately to the market. Well, what happens if you hit a snag? So all of this is extremely complicated and expensive to get through this whole process. And that's if everything goes perfectly. The problem is that nothing ever does go perfectly in this world. There's lots of challenges in developing new technologies. And when you're doing research, this is why we do research. We don't always have the right answer from the beginning. If we did, it would be easier and everybody would do it. And so there are a lot of things that you learn as you go through the process of developing new technologies, as you start to do your testing on the benchtop, as you start to do your testing in animals, and then even when you start to do your research in humans. And so if there's a problem, you have to go back to the drawing board. And there are a whole lot of steps that have to be done in order for you to make sure that you're figuring out the problems that there may have been and why, and to ensure that you can address those problems safely. And then you have to redo all of your testing. You have to redo your negotiations and your communications with FDA. You may have to redo your communications and negotiations with CMS. You have to develop a new IDE, new submissions, new clinical study sites potentially, more training, more education, and all of that costs a lot of money. So this is a quote from the Stanford Biodesign Handbook. And really, this is something that I think anybody in tech development should understand. But I'll just read it. Human device testing is at the pinnacle of med tech innovation process. The stakes are high. It takes 2 or 3 years to perform patient recruitment, enrollment, investigation, and follow-up. Costs can be as great as $100,000 per patient study. Regulatory and reimbursement issues are hanging in the balance. The credibility and commercial viability of the company is on the line. And most importantly, over all of this, patient lives are in the innovator's hands. And that's really heavy when you think about it. But that's what it all comes down to. So I want to talk just quickly about funding. And really, I'm bringing this in here at the end to bring up some issues. Really, funding comes at the beginning. It has to come immediately or you can't go anywhere. You can't do anything without money, particularly on this side of the world. And so there are options of personal funding, using your own money, academic grants, government grants, business loans, convertible debt, angel funding, venture capital. You've probably heard of all of that. And the answer is really, you need all of it. You need as much as you can get. And just as an example, this is some of the examples of some of the funding that we've been fortunate to lock down over the years. It really started out with a great idea and some founders who were willing to put up some money on their own to be able to get this to move forward. But beyond that, we've been very fortunate to secure some academic grants and then some government grants. As you can see with the NIH and the National Science Foundation, we've been able to secure about $6 million from that. These are high stakes grants. They're not easy to come by. It takes a lot of work and it takes a lot of time. Even if you're successful, it takes a long time for this money to come through. It doesn't just hit your bank account the next day. We've had business loans with our local NC Biotech, and we've been able to secure convertible notes or cheaper equity funding. And we've secured close to $6 million with that as well. And to date, we've been very fortunate to not have to move to venture capital at this point, but there are other potential options for funding and strategic investments as well. But to date, this journey has required at least $13 million for us to get to this point, and the journey's not over. So I just wanted to mention quickly about strategic partnerships and acquisition. This is something that, particularly in the medtech device world, is a typical goal. Nowadays, there are a few very large medical device companies out there that control most of the med tech. And when you're a small company starting up, in general, the goal isn't to become all powerful and buy up all medical technologies and become one of the behemoths out there. It doesn't tend to work that way. And so in general, one of the main goals is to develop a good product till it gets to the point that it piques the interest of one or many of the medical device companies out there, so that they can come in and they can help further develop the technology and bring this to patients with more assets at hand. And so, unfortunately, nowadays, things change over time. And nowadays, there are fewer early acquisitions from these big companies. And why is that? And it's because the big companies don't want to deal with the regulatory risk and those distractions, because nothing is a guarantee. And unless it's approved and cleared by FDA, it's not going to be delivered to patients. And if it's not approved by FDA, it's not going to make money. You can't sell it. You can't use it. So there's no value. And so how it goes nowadays is that the onus is on the small biotech companies to foot all of the funding initially, on all of the risk initially, to be able to get it to the point that it is a promising and solid technology, so that a large company could potentially acquire your company and or invest in a partnership. But unfortunately, the small companies typically are run by less experienced folks than are on large companies, and they have less resources at hand for sure. And so because of this, and I've personally witnessed, and I'm sure many of you have witnessed, that many good technologies die. And when they die, they often do not become resurrected. In some cases that can happen. But when a technology dies, it's oftentimes gone. And I think that's really sad. And I think that currently where we're at is the current model does not benefit patient. It does not benefit patients the most. It tends to benefit companies the most. And that really isn't aligned with where we should be going for caring for our patients and bringing new technologies to bear. And so what if the government started to subsidize big industry to invest or acquire in small companies early for promising technologies? That could start to shift some of the burden and some of the risk from the big companies, but allows promising technologies to move forward. And so when you run into a snag in a clinical study and you need to redevelop, that company can potentially weather that snag, whereas many cannot. And then it starts the death spiral to the end of a technology and the end of a company. So in summary, I think innovation in end-stage kidney disease and dialysis is continually underrepresented and underfunded when you compare to other respects of medicine and technology. I think there's more funding for kidney care research and development that's required. I think that we need to continue to maintain a heavy focus on patient safety and satisfaction. This is something that it should be at the core of all development for patient technologies. We need to continue to push for home therapies, and that's not just in the hemodialysis space or the kidney care space. That should be in all respects of medicine. But we have a unique time and a unique potential here in this period of time to push more patients to home, so that we have patients that are more comfortable and that we have patients that have more effective and better outcomes. We think that with our technology, we think we have something here, right? I mean, we think it's a great idea. We've seen it help patients directly already to date in our clinical studies, but the road to success is long and it's hard, and funding really oftentimes is the rate limiting step. FDA and CMS must continue to have strong expertise in the clinical space, and this is not always the case. And so it can be very difficult for you to articulate to these agencies about what are the important aspects of care and outcomes in the particular field that we're talking about. What are the most important outcomes? And this is how your clinical trials are developed, and these are how your benchmarks are set. And so I think it's imperative that FDA and CMS can have strong expertise in the clinical space when they're reviewing your technologies. think furthermore, it's very important for FDA and CMS to collaborate and to align on the requirements for clinical studies. Currently, these two agencies are in silos when it comes to requirements for clinical studies, and it can really cause a lot of confusion and a lot of delays and a lot of wasted money while trying to manage these issues. And I think, again, considering federal subsidies for the private sector for early investment or for acquisition for promising technologies could be a potential solution or a potential way to improve innovation for patients to bring safer and better technologies to bear. So I just want to recognize the team. This is a long and difficult road, and it takes a lot of people and a lot of dedication and a lot of effort to get to where we've come, and the road's not at its end. We still have a lot of work to do, and we're still very dedicated to helping as many patients as possible. And I really want to thank the organization for thinking of us, bringing us into this meeting so that we can hopefully spark some interesting conversation and debate around how we can continue to make things better for patients, how we can improve the potential for more small companies to continue to innovate and improve. Thanks very much.
Thank you, Shawn. And thank you to all of our panelists for sharing your time, insights, and expertise in the importance of advancing innovation and supporting patient care, choice, and access. Thank you.